Good day, I'm Aldrin Dogan and I'm going to present to you my ARCDES9 thesis proposal. As human beings, we have been fully aware that we are coexisting with different species in the planet. We have been growing with them, and this system of coexistence with other species is called the ecosystem. It's the functional unit of the biosphere. While the variety of species in the ecosystem is called the biodiversity, these are two terms that are fundamental factors in our planet. With that, I'm now going to present to you my thesis topic. My thesis topic is Hybrid Biotechnology Garden, a research conservation facility for endemic plant functional diversity through meteorological architecture. Let's go one by one in the process that I have taken into. This is the ecosystem energy pyramid. This diagram shows the transfer of energy in the ecosystem. Scientists have been telling us that plant at the foundation of this pyramid is the best barometer in knowing the status of the whole ecosystem. These are the biodiversity hotspots around the world, and Philippines is one of them. Philippines is a mega biodiversity hotspot containing 70% of the world's flora and fauna. Philippines have a 60% endemic percentage of plant families, meaning that 60% of the species that you can see here are unique in the country. With that knowledge, IUCN has stated that biodiversity has been affected by anthropological activities. They have listed the top three biodiversity killers, namely overexploitation, agricultural activity, and urban development. The anthropological activities has been monitored by the Kew Gardens. This table has two rows showing data before industrialization, before 1900, and after industrialization, 1900 to the present time. This table tells us that there are 59 species that went extinct in 100 years. There is a 111 years decrease in seed plant life expectancy and the 221% increase in seed plant life extinction rate. This is the map of plant extinction. But since extinction is a normal phenomena, they have also mapped the rediscovery of species, meaning how would species bounce back from extinction. Philippines, based on the map, have 41 to 79% extinction rate per million species and have a poorly 1 to 2 discovery rate of rediscovery per million species. Philippines is experiencing a mass extinction without mass rediscovery of species that shows problems of the conservation status of the country. In terms of the conservation status and the strategy that is used, the global strategy for plant conservation is stating that 75% of plant species should be extracted to ex situ collections. 20% of the genetic resource from those ex situ collections should be available for restoration programs. What is a plant genetic resource collection? It is a germplasm. It is a collection of germ cells or a gene of a whole culture. The facility that stores this is called a germplasm facility, and that is where you study the genetic resource. There are types of germplasm, starting with in situ. In situ germplasm is the use of legislation to protect a geographical region. In the pictures, there is Region 2 Batanes Group of Islands located in the northern part of the Philippines, which is the top 1 biodiversity hotspot in the country. Region 8 Summer Natural Park, which is the top 2 protected site in the country. The next type of germplasm is ex situ. Ex situ germplasm is the protection of species outside its habitat. On the right side, you can see the picture of Eden Project in the United Kingdom. There you can see the different biomes with different types of climatic conditions. They extract species around the world and let them proliferate inside these biomes, then later reintroduced to the countries it was extracted from. In terms of local example, we have University of the Philippines Los Baños in Laguna, practicing this kind of conservation strategy. UPLB was built at the foot of Mount Makiling to extract different species near protected site and also other species in Luzon. They are using genetically modified organisms to improve plant life expectancy. This is the status of Philippine ex situ collections. These are our gene banks around the country. In terms of in situ collection status, we have different parameters in trying to measure the performance. First one is coverage. Coverage is a biodiversity that is covered by the government in terms of protection. In the country, we only have 11% out of the 27% biodiversity hotspot that is protected. The second one is management effectiveness. It is the existence of a legal management plan to systematize the protection of the site. 40% out of the 80% of the protected site have detailed conservation plan. The last one is the overlap of biodiversity. It is about the mismatch between the protected areas and the key biodiversity hotspots, meaning the government protecting the proper places or the proper species. We have 64% mismatch. This shows that we are lacking accuracy in which should be protected by the government. This is the summary of the problems in genetic resource management. Philippines have fragmented out of coverages in need of improvement in terms of genetic resource management. With that, this is the statement of my problem. One of the main root causes of the rapid plant extinction in the Philippines is due to inadequate biodiversity coverage of facilities and fragmented strategy for biodiversity conservation. The research goal is to propose a modern genetic conservation facility that will be a common venue for hybrid conservation strategies and an improvement on endemic plant biodiversity genetic coverage. These are the research questions in line with the research objectives. The first one is how to provide an effective typology that would improve the genetic coverage of our conservation. Our objective is to provide this improvement by combining in-site and off-site conservation strategy through conservation-oriented restoration. What type of spaces the scientists need to study plant growth? We are going to use case studies of germplasm systems and utilization to look at the different processes of plant growth experimentation. What systems and protocols does scientists execute to do plant growth studies? We are going to look at case studies of the process of germplasm and biotechnology systems in order to identify what scientists have been doing. How to provide a collaborative social and scientific space that will improve cooperation and information sharing between scientists and lay people. I'm going to use ecomimesis design to focus on spatial studies of functional diversity. With that, these are my case studies. The first one is Eco Boulevard in Vallecas, which focuses on the process of urban recycling operation with the principles densification of alignment of trees, reduction and arrangement of wheeled traffic, insertion of an air tree social dynamizer on an existing urban area. The design process is created through evapotranspiration. The design has three layers. The first one is the technological part, which encloses the plant diversity, which is the second layer. Then at the bottom are the humans under the active space. There is an interrelation for humans to plants then into technology, which creates the process of evapotranspiration by the plants absorbing the sweat and heat energy from the humans. Then through the structure, the plants regulates the temperature below. The theory that is used in this architecture is ecomimesis design, which is the programming of homeostatic functional diversity, with the principles of homeostasis, spatial efficiency, succession, biodiversity, ecological cybernetics, and biogeochemical cycles. This focuses on the processes of designing the living part of the ecosystem. The next case study is Jade Eco Park, focusing on the atmospheric quality of the artificial environments. This is the process of the designing the park utilizing the southwest wind velocity to map the possible cool places. In this pocket of different temperature is where the architect places the intervention. At the last map is the effect of the architecture, where there is an increase in humidity. This is the architecture, which are small insulation of which changes the humidity of the surrounding area. The architect has two considerations, which is the meteorological through the environment and the physiological through the human metabolism. The theory used in the case study is meteorological architecture, which focuses on heat and light source, color reflectance, relative humidity, insulation, ventilation, and dual airflow, which focuses on the abiotic or the non-living factors of the ecosystem. The conservation strategy that will be synthesized with the two theories is the conservation-oriented restoration. The conservation-oriented restoration integrated the two conservation strategies we discussed a while ago, creating hybrid typology, which is called the interquasi germplasm. An interquasi germplasm is built on the buffer zone of protected site. The protected site will be a catalyst through the interquasi germplasm. The hybrid germplasm will be extracting 70% of the genetic resource, then will be reintroduced again into the protected site, while 20% will be propagated into different suitable protected zones in the country. The germplasm have three phases. Plant acquisition, which focuses on the quarantine and culture control of the plants. It is to identify if the plants have pests or invasive qualities in the surrounding ecosystem. The next one is plant conservation management, which focuses on the growth of the plants, both in horizontal and vertical structure. The last one is the germplasm utilization, which focuses on how you manage the data and information that you get from the germplasm. We will now put our architectural theories. What is in between the theories and the conservation strategy? Here is my theoretical framework, which is the synthesis of the two architectural theories creating the experimental biomes as the synthesis. The experimental biomes would be in the middle. The experimental biomes would consist of the three types of spaces: artificial, semi-natural, and natural spaces. In the conceptual framework, we would now put the conservation strategy on top of the two theories creating my thesis, which is the biotechnology garden. The biotechnology garden is run by three principles overlapping between the conservation strategies and the theory. With the meteorological architecture and the conservation-oriented restoration is bioatmospheric morphogenesis, which is the morphing of shapes of architectural spaces through the atmospheric condition needed by the biotic factors. Next is the overlap of ecomimesis design and conservation strategy, which is the simulation of plant growth in the controlled space, along with the experimental biomes, which is the architectural thesis of my ARCDES9. This is my analytical framework, the summary of the process I have taken into, the analysis, and then the synthesis, which is the synthesized hybrid biotechnology garden. Let us now go to the site. These are the parameters in choosing the site. It should be a priority for conservation. It should have an existing protected area. It should have an existing buffer zone. It should have a big zoning area. This is the top 5 biggest protected areas in the country, with Region 2 Batanes Group of Islands as the biggest area, but it doesn't have a buffer zone due to its seascape structure. Region 8 is the next choice in terms of the site, which is the second biggest protected site, with 437,220 hectares of area. This is the map of Region 8, which is Eastern Visayas. These are the protected sites inside the region. These are the top 3 biggest protected sites in Eastern Visayas. Summer Natural Park is the biggest one, and also the Center for Plant Diversity. This is the map of Summer Natural Park, spanning to the whole island. The green part is the protected site, while the yellow part is the buffer zone of the protected site. The buffer zone acts as a barrier that helps in conserving the natural park. This is the macro site analysis. We have the provincial boundaries, the contour map, the wind map, the landslide hazard map, the rain gauge map, the flood hazard map, the storm surge advisory map. These are the 3 biggest cities and municipalities inside the buffer zone. Calbayog City is the biggest one. We are going to build the structure there. This is part of the buffer zone that we are going to build on. This is the site. This is Osmeña Samar. There are 2 existing barangay in the site, which are Barangay Bagong Lipunan and Barangay Pato. The site is a valley formation. This is the topographic map. You can notice that there is an increase on altitude on the bottom right part. The increased altitude is the protected site, while the lower elevations are part of the buffer zone. Here's the site-specific boundary. This is the natural areas of the site. Here is a topographic model. You can see the valley formations in the site. These are the spaces that will be built in the site based on my thesis. We have the artificial, semi-natural, and natural spaces. The experimental biomes will have a different simulation biomes, which is the innovation of my design, which has tropism biomes, which is the nature of plant to go towards light, water, and the source of gravity. We also have the soil and water simulation biome, which is observation of the growth of plants depending on the quality of the soil. Sound simulation is the nature of plants to react towards the sound. High altitude biome is for plants that grow on high altitude places. The ecological corridors is the connection of semi-natural to natural spaces and artificial spaces. The collaborative school for plant nursery is aimed to socialize the awareness that is created for plants in the architecture typology. This is the major vehicular circulation. Area for vehicular circulation, the sole objective is the reduction of wheel traffic. Walkable area, objective is to increase the walkability in the site. From my experimental biomes programming of my thesis, we have the first type of zoning space, which are the artificial space. The next one is the semi-natural space. The third one is the natural space. Here, We provide ecological corridors to facilitate plant growth from artificial to the natural spaces, and also the Center for Social Interaction. We will provide plant acquisition facility, which is included in the phase 1 of the germplasm utilization. We will provide plant quarantine and records keeping spaces, which is part of the phase 1. The next one is plant conservation management facilities, which is included in the phase 2 of germplasm utilization. We will create analog growth simulation biomes, which is the tropism biomes for the growth study and simulation of plants in the nature of endemism of plants around the site and eastern Visayas area. This is the endemic arboretum, which will be a collection of endemic trees around the Visayas area. An endemic botanical garden, which also be a collection of plants in the Visayas area. This is the legend. This is the first architecture and the phase 1 plant acquisition. It is designed for the acquiring to identify invasive endemic plants. This is where you will know which plant is good for natural growth and good for artificial growth. The list of spaces will be vertical gardens corridors based on ecomimesis design and the active leisure space below it yielding the evapotranspiration process that would help the plants to grow. Vertical endemic garden on top and laboratories below. Along with this is the unlabeled genetic resource storage. It is unlabeled because it is the initial plant acquisition. The next design process is the phase 2 or the growth biomes. We will start with gravitropism, which is the nature of plants to go towards the source of gravity. We have 4 configurations starting from the positive gravitrophism, neutral gravitrophism, and negative gravitrophism, and high altitude gravitrophism. It is divided into modules of plants. We have high altitude plant modules and plant growth towers, and grand observation circulations for scientists to see what is happening in the plant growth modules. We have an exterior wall and an interior wall with a buffer zone in between for the utilities. This is the elevation on the left side. The next one is negative gravitrophism. We have a light passage below with most of the plants underground. This is on contrast with the high altitude biome. Without the absence of light, the only thing plant follows is the gravity. We have underground plant modules and circulations for observation. This is the positive gravitrophism, which is the standard height for plant growth vertical propagation. We have interior and exterior walls and ground observation circulation. This is the neutral gravitrophism, which is divided into 2 sides. One is below and above ground. We can see here the contrast of growth of plants in the influence of gravity and light. This is the phototrophic biomes, where we study the nature of plants to go towards the light, answering the questions of what would do if you increase the intensity of light, and the fourth one with ambient light through the perforated controlled wall. This goes with the topic of genetic modification and genetic mutation that will happen in these types of condition. This blue light irradiation, which is the blue configuration of phototrophism. This is the photoperiodism, basically the nature of plant to go towards a cycle of night and day. There are plants who grow in the winter solstice due to its exposure in the nighttime. There are different openings, a 12.5% opening, 25% opening, 75% opening, 87.5% opening in the interior wall. The interior wall is rotating. The rotation of the wall is controlled by the scientist. The rotation would yield the controlled environment to know what is the critical day length for short day plants. Meaning, if the plant is not exposed to light at a specific duration of time, what would happen to them? Would there be a genetic mutation or a new phenomena that we don't know yet that we can know in this type of space? Sound on plant at atmospheric transfer. Sound has been studied and has been observed to increase growth and cell wall fluidity in plants. There is a functional diversity at the bottom, then sound is coming from the sound amplifier. If you have an atmospheric transfer, there is also hydroponic transfer. We have 2 observation desks, one on top and one at the bottom. On top is where you can see the flower, the blooming of the plants, and at the bottom is where you can see the nature of the roots towards the sound. We have the functional diversity on the middle, then the water tank axonometric on the right side. This is the soil and water. Here you can see the 2 layers, which is the experimental topsoil and the experimental mineral soil. There are plants that when you plant them on Mindanao, then you extract them to the Luzon area, it changes color. Research objectives and design solutions. The planning site criteria and placemaking through conservation-oriented restoration of the facility within the chosen site creates a larger coverage of protected sites in the Visayas and Mindanao region. Through the provided hybrid architecture spaces, Questions about conservation of our endemic plants can be answered. The planning of the facility is based on three layers of types of spaces that follows protocols of germplasm conservation: natural, semi-natural, and artificial spaces. The collaborative social space through School of Plant Nursery and ecological corridors will give experiential activity while giving awareness and accessibility to biotechnology and information. This is my thesis. Thank you for listening.